So my friend and colleague Christophe Lambert, he did his undergraduate. He started at University of Calgary and then moved to Montana State University. After that, he went to Duke University and did a master's with a thesis looking at algorithms for particle assembly. And then he did his PhD at Duke as well in computer science. And that was really looking at algorithms for biophysics as well as non-parametric methods. And then after that, he was working for a hedge fund, apparently doing some algorithms for people to make money. And so once I get some money, he's agreed that he'll show me how to invest that. And so after that, he went and started his own company. So he's the founder, president, and CEO.
Well, not CEO anymore, but...
Well, let me finish. He was the founder, president, and CEO of Golden Helix. And I actually... we've written some grants together. He's been successful with some SBIRs and has a nice portfolio of funding from both federal sources as well as the private sector. And one of the things that when I first met Christophe that really impressed me about him is that he was running the company and truly immersed in business, but because he was working with a lot of academic partners as well as pharmaceutical companies, he has a prolific publication record and an excellent funding record in that venue as well. And so we actually met at Santa Fe Institute. He gave a talk up there. I think it was, I'm bad with time, it was 2 or 3 months ago. And he was telling me that he had stepped down as the CEO of Golden Helix, and he was interested in moving into academics. And so we have been pushing him hard to find an interest in UNM, and I think it would be a great asset to our team. He fits into multiple silos. I couldn't really put him in a category. He does informatics, he does predictive modeling, he's an expert on theory of constraints. But I think he's something that could bridge a lot of different projects and groups that we have here at UNM. And I'll let it go at that. Welcome, Christophe.
Thank you. Thank you for having me.
Let me just say that I think it works best with the overhead lights off and let's see.
Sorry. What do you think? Well, you don't want it lights off.
Yeah.
A little dark. It's okay. Hopefully, we'll stay awake.
That'll be up to you.
Yeah. We'll see. And currently, I'm also teaching a semantic web course at Montana State University, which is an interesting thing. Actually, I'm speaking at the Veterans Administration at noon today, and I'll talk a little bit more about some of that work in the context of healthcare. I thought it'd be useful to give you sort of a visual of some of the things I've done in my career. And on the tops, kind of the things I've done, and on the bottoms, sort of the systems and thinking approaches that I've picked up along the way. This talk's, in a way, a meta talk where we're not just saying, well, there's a science, but thinking about how do we go about doing our science to get more out of our time and energy. And so, across all these years, of course, I've been doing, as DG said, computer science, algorithms, information theory, and that shapes my worldview. As a company that serves the academic world, most of our customers are in government labs and academic research organizations, as well as pharma and biotech. We serve hundreds of people who do research, and so we're kind of an enabler for other people to get their publications out. And so I've had this vantage point of serving many, many different academic groups where you can start looking at the common issues, common problems, and various approaches to systems thinking have been enormously helpful for me. And it kind of came out as, 1998, I found Golden Helix, and I'm an academic. I don't know anything about running a company. I actually thought some other guys were going to join me and I read all the management stuff I could and found the flavor of the month stuff didn't work. And as a scientist, I wasn't satisfied till I came across some systems thinking disciplines that gave me, if you will, a scientific window into the way organizations work so that I could then improve it. And so theory of constraints, system dynamics, I'll I'll be talking a bit about, I'll be using some of those to frame the way I think about the organizations, think about our healthcare systems and so forth. So I've done, of course, you know, if you look at my resume, there's all this stuff on genetic research, worked on dozens of projects with customers in doing GWAS, copy number analysis. And then there's a whole period both at Glaxo and at the earlier stages of Golden Helix that I worked in chem informatics, developed software for that, and did drug discovery efforts. So the sort of the way I'd like to set the stage is, imagine if we could actually systemically improve our research productivity tenfold. And you know, we sort of have to, what do we mean by productivity? And then is that even possible? And what are really the obstacles and blocks to doing that? And can we remove them? So if we look at the world of engineering, we're familiar with Moore's law, exponential drop in prices, and exponential increase in speed and compute power that has been sustained over decades. Computation, started way back. And of course, we see it in sequencing with the high throughput sequencing has gone even faster than Moore's law. There was a place here in where they started using silicon-based technologies that probably we piggybacked on some of Moore's law, and there was a real inflection point in our speed of doing sequencing. And so we've seen this amazing acceleration in R&D. And then if you look interestingly at one measure of research productivity is publication output. And so from 1985, I took the NIH Project Reporter data, and you can take the grants and you can take the publications that came out of various grants, and you can see that an interesting thing has happened. The number of people working on projects has increased, and so we got more authors per publication. But if you take the total number of people total number of publications divided by those unique authors over time, it's actually been dropping. And so the units of publication, if you will, per researcher has been dropping. That is, if you were to have 5 people on a paper, you'd give a fifth of a paper to each person. So in this metric, even though we know the total number of publications has been accelerating, there's more people doing them too. And the dollars per paper is about at $350,000 to actually get a peer review. If you take the total NIH dollars divided by total NIH peer-reviewed, the peer-reviewed publications. And so there's some interesting, each of us say, $350,000, I get a whole lot of papers for that. Well, we're also dividing by author names, so this money's spread over everybody. For those of us who've worked in the field of drug discovery, we're familiar with this long slide since the 90s in new molecular entity production, so getting drugs approved and so forth. Despite continuous, I don't know if it's exponential, but it's certainly a steep increase in research expenditures, the actual output has been dropping. And the biggie, which is if all this research is directed towards life and health and producing life-saving medicines, helping us live longer and having more productive life years and so forth, we have been increasing life expectancy, but only linearly, and we get about 1 year for every 5 years of research. And it'd be great if we're all born 100 years from now, because it would be. But as it and however, and if you look at expenditures, this is perhaps a little hard to see at this resolution, but this is percentage of GDP versus life expectancy. And so for all the expenditures, the U.S. is around 16-17% of our economy is spent on healthcare. And the Singaporeans are spending around 3 or 4, and living just as long, at least as of 2007 when this data was collected. And at the high end, we know we gain a lot through worldwide on things like sanitation and so forth. But at the high end, for octogenarians, people who are 80, we've only gained 3 years of life over a century. So putting more money, lots of smart people are working on this. Something in the system is not producing, exponential improvements for us in terms of life and health. A word on exponential growth. The key to understanding it structurally is the thing that is growing produces more of itself. So we're familiar with that with population growth. People have more people by having babies. Remember when our bank accounts used to give us compound interest? Now the rates are so minuscule. But money makes more money. And so there's a phenomena, why do you see exponential growth in knowledge? Is knowledge itself begets more knowledge. And it's not just the computer revolution where computer speed has been plowed into enhancing, say through CAD systems, better computers. You can look at fields like optics from the 1600s. There's been an exponential growth in telescopic magnification. So there's also resources that come in to fund the improvement of technologies. So Arthur Conan Doyle probably first represented this idea that knowledge is a self-growing process. So we've had this exponential improvement in computation. We know our instrumentation is getting more and more sensitive. We look at microscopy, we look at NMR instruments, we look at high throughput screening, the scale up that we've had in terms of number of compounds we can test. We've had these exponential growths. We've even had exponential increase in expenditures until about 2003 with NIH funding, and PhDs and knowledge. And there's plenty of economic incentives to, we don't want to die or be sick. And yet, unlike engineering, the biomedical system hasn't yielded this exponential bang for our buck. And in particular, I'm going to like to talk a bit about why drug discovery development is decelerated. My VA talk later today, I'm actually going to talk about healthcare in general. And it's really about the complexity of biology and the challenge of managing that but I'd like to use drug discovery as an exemplar that the industry's been trying really hard to get more out of it, but hasn't been. And so I'd like to drill into that. Maybe not all of you are interested in the details of drug discovery. A lot of you, though, are actually work actively in this field. And then we'll come back to the research process and look at productivity there. So this decline, everyone's been bemoaning it for a decade. We've been bemoaning the slide, and it's approaching 20-year decline. There was this President's Council report. Eric Lander and others were saying, we need to spend here to fix this problem that we're not having, more life-saving drugs. Unfortunately, while I had plenty of places to spend money, there was not a diagnosis of the systemic cause. Now I'm going to have some hypotheses about it. I could be wrong. But what was interesting to me is here's a lot of top flight people who are setting policy and so forth. And before taking action, there wasn't a diagnosis. And so as doctors, we would want to diagnose the patient before prescribing medicine, and I think there is an implicit diagnosis probably that we're just not spending enough here, but as the data is shown on the spending doesn't necessarily produce more output. I wrote a book chapter back in 2006, and what was an interesting thing that came out of this analysis, and this is a cause and effect called a current reality tree, where you can take the problems in a system, and the amazing thing about systems is they tend they broaden out and then they lens down to a core leverage point where you can actually tackle the problem. And in the case of pharmaceutical R&D, there's a couple interesting things, but the key insights were locked into blockbuster mode. You focus on the same disease areas, and then to beat all previous comers, it's a monotonically increasing sequence where you're trying to beat, in terms of safety and efficacy, the previous comers. So then that becomes, you have to be more and more innovative to beat everything else that has come before you. And then, of course, the cost equation that comes in there with all the failures of trying to beat it creates this idea that we've got, you know, $1 billion+ in R&D to get a drug. And there's also some interesting structural things with regard to the whole shift towards emphasizing generics and so forth. The core problem is it just takes enormous cost to do all these clinical trials and so forth, and we're failing with really high percentages at these very expensive stages. And so Deming showed that errors in a process magnify over time, and so the best place to intervene in a system is to reduce the problems at the front end of the funnel. And so the failures that occur essentially have to do with our in vitro models don't map to in vivo outcomes. So there's this gap between my experimentation and what happens in reality, and we're trying to, you know, model a living system in a test tube or a 1536 well plate. But the reasons they fail are for ADME, tox, non-specific binding, or no therapeutic benefit. And the question is, can we address these failures at the beginning of the process? So one of so I built a product called ChemTree that I'd actually developed as a graduate student, beginnings of it at Glaxo Wellcome. We patented some stuff on a basically predictive modeling for structure-activity relationships, but it had to be used in the context of a process. So you sort of have this hammer, but unless you wrap around it an appropriate system for drug discovery, you're not going to get the benefit. And if you look at traditional drug discovery, there's this great technology called high-throughput screening. Some would say not so great because, hey, what have we gotten out of it? They might use some CombiChem or QSAR to expand hits. You would do this high-throughput screening at the front end of the funnel, saying, I'm going to throw millions of compounds to see what sticks, what binds to a target, and then typically in pharma companies, throw it over the fence in a linear discovery process to medicinal chemists and say, okay, this binds a target. Now medicinal chemists who can build one compound a day if they're really good, often one a week, go make this safe and effective in human beings. And so we've now created a choke point where they we're at a local optimum in terms of binding against the target, but all the other dimensions that matter for in vivo effectiveness, is it safe and effective and good ADME and tox properties, our in vitro models, we haven't applied that automation to all of the dimensions, and so it's no wonder that we fail for all these dimensions that we haven't used our technology to tackle. So the approach, and the interesting thing is this approach we were selling a paradigm shift with one piece of the paradigm, say, here you need to change your whole process. Very difficult to sell, and very few, in fact, nobody has ever done this entire thing, which is what you want to do is have great in vitro models for as many of the things that you can try to predict in vivo. There was a company back years ago, and we did some work with them, called NovaScreen. They were later acquired by another company and another company. They had several hundred assays going at a time for all sorts of ADME and tox endpoints. Unfortunately, it would cost you thousands of dollars to run one compound because you had 500 assays. Plus, they weren't the people they were selling to were using this late in the discovery process. So saying, okay, it's past all other hurdles. Let's go send it to NovaScreen and then run it against their panel for liver toxicity and so forth. So the price point for this made it prohibitive for even medium throughput or even low throughput screening. But what I would advocate is if one could get the cost down, you would basically set that up at the front of your process. Maybe not high throughput screening against 300 assays, but medium throughput screening, have a machine learning model, which is the hammer that we built, to screen to screen against all these assays, climbing the multidimensional surface towards better, not only potency against the primary, but all the other dimensions. And then there's no machine learning algorithm that can extrapolate very far beyond what you actually are giving it. So you essentially have to have a good diverse set that somehow covers the space. You learn from actually doing screening, but then you do an iterative learning process where you build a model. You then screen from a vast universe of compounds, and most collections are limited by 5 to 10 million compounds, whereas the search space is 10 to the 60th potential compounds. So the power of CombiChem is that you can virtually create compounds in a computer, screen them against with a virtual predictive model, and then build the ones that actually are expected to have effectiveness, and then you iterate this process. And so you use the power of CombiChem to enhance your search space. Ideally, we automate this, so you'd have a robotic automated drug discovery process, and take this thing forward. And now, if it didn't work, you might have to start with some other diverse compounds and search other parts of the space, but then take this through animal and human trials. And then all the information you learn from animal and human trials, including why not start with all the drugs that have ever been tested in humans and put that in our process as well and run them against all these screens. So we can slowly learn on the smaller end of the things we're actually testing in vivo. We built a product to do that, some literature contributions. What was interesting is we could actually even do some directing of the medicinal chemists with the machine learning to the specific parts of the compounds that were the business end. So we could actually say this is what matters in the ones that are successful, and this is the parts that actually might detract from activity. So you don't necessarily remove the chemists from the equation, you give methods to empower them. There's a whole lot I've done for 2 decades on machine learning, as DJ said, in finance, and then in chem informatics, and later in genetics. The key is any one model is only going to be so good. And much like that TV show Who Wants to Be a Millionaire, and you ask the audience to vote, you get a better response by having, if the audience knows something, if it's not random knowledge, and there's some statistical independence. And so there's an approach with building good but randomly perturbed models that you can get an average prediction that's much better than any one model. Again, here is where we could do some stuff to actually, this was a known example with MAO inhibitors where we could point to the business end of the molecule and it would corroborate what was known about how this molecule operated. Also, we built some special stuff for this idea of multidimensional prediction, where you could look at all dimensions simultaneously in a predictive model. And this was looking at the NIH disease screen. And without going into the details of this, you could essentially, with a modest size sample, build a good predictive model that could get most of the active compounds in this particular screening library with much fewer actual total tests by this iterative learn, test, repeat. So it's really stochastic optimization. And there's no perfect algorithm that's perfectly predictive, but there's no human being that can know immediately what to build either. And so if we can use stochastic search to speed up the process of searching that multidimensional space, we can get actually orders of magnitude enrichment at the first stage and even subsequent stages. So what came of all of this? Well, our customers confirm, yes, they could get these increases. And of course, we're not the only people who've done this type of predictive, but we're very early in the field. my colleagues at Glaxo Wellcome and I published some of the first papers on using recursive partitioning for structure-activity relationships and predictive modeling. This product's actually embedded in a meta-drug product at Thomson Reuters. We still get royalties from it, and they can they add the metabolites of compounds as well as the compounds themselves in their predictive modeling. One of the life lessons here was selling. You know, when your customer has to buy a paradigm shift in order to get the real value out of what you're selling, very difficult sale, and so selling just a piece of it of that paradigm shift, you know, is is a fool's errand, let's say. So, but a company that had a similar process, didn't have all those stages. They actually licensed, Neurogen licensed a similar sort of end-to-end thing to Pfizer for $27 million. Even then, if you go look at the history of it, it sort of sat on the shelf and the paradigm shift didn't occur. So we get into these questions of we want to do something better, but we're having to change the existing order. It's often an uphill climb. So anyhow, I would love, so part of my desire to consider working in a drug discovery and also potentially integrating with genomics and so forth is saying, you know, it's one thing to get on the soapbox and just be the computational guy saying you need to change your processes, and, you know, another to be able to roll up your sleeves with colleagues, hey, you know, can you buy into this idea? And this may not be a complete solution, but let's think about that process, because what we've been doing thus far hasn't served us well. How do we do it and do something completely different? And we don't have 300 medicinal chemists. We're going to have to do something smarter and automated. And so I'd like to return to examining bioscience research. So what's systemically different between the engineering and biosciences research? If you think about the cycle that we do in science, we're probing and testing, and we build a mental model of reality, we reconstruct an experiment, and we try to falsify it, and we update our mental model, we do our experiments, and there's basically a loop. And this OODA loop, for anyone in the military, there's a guy named John Boyd who actually I think it was General Powell said that John Boyd was essentially the spiritual father of the Desert Storm Initiative. So his whole theories on warfare, on how to have a faster cycle time than the enemy, basically are what created this shock and awe, incredible rapid victory in Iraq in the 90s. And so, but it was actually it wasn't just a warfare paradigm, it's a paradigm for how it is that we iteratively improve our mental models. And this happens at an individual level, but also happens institutionally. Our research cycle time in pharma is actually constrained by a security need, i.e. we can't treat people like laboratory animals. We've got to be extremely careful. We can't just go to in vivo testing, you know, on people. And so, you know, drug development, in part because of that, is a 14-year endeavor. One thing we don't have that it's nice to see there's some good work going on here, for instance, with Jeremy on modeling, you know, building really working models of systems, is like in computer design engineering, we can build very solid models of what we're actually creating in reality. So much of what we do in biology is unobservable, and as a result, and it's also as a result of probably the emphasis on biology as opposed to sort of engineering. The models that we use to represent reality in biology are often cartoons, right? If you look in a paper, and here's a nice picture of how something works, but in terms of is it an actual compute model that you can test rates against, test the flows of materials, and so forth, we tend to be impoverished in that area. And so there's a field called system dynamics that I've spent a lot of time working with some of the world's experts, and there's people out of MIT that do this, where you can turn that cartoon, if you will, into a working model that you can then say, okay, if I intervene here in a system, what's the expected effects? And the key, though, is getting behavior over time data. Another thing that's often missing in a lot of our studies of biology is we kind of do correlations. We take a snapshot in time, or we do before, after. The real power of looking at biomarkers over time in years to come is that we're going to be able to start building causal models. And in order to understand cause and effect, we really need time. And if you think about it, so then this is something I'm going to expand more in my VA talk later, but complicated versus complex systems. Cause and effect is changing in biology. You know, a circuit is pretty much a fixed business. In software, we don't allow self-modifying code because we know it becomes unmaintainable, but we're actually self-modifying systems. And so there's a key thing, again, I'll talk about it in my other talk, from cybernetics, which is a wonderful field, came out of operations research in the 40s, and there's an active community in it, but there's a key concept called requisite variety. And it says simply, in the simplest way you could put it, if you're trying to control a system, the agent that's controlling a system needs more degrees of freedom than the thing being controlled. Okay? So if you think of the degrees of freedom of a drug, it's pretty much designed to hit one place on the system. You think about these complex systems that have many moving parts, we can fix one of the variables, and if there's more governing variables to that disease than what a single molecule can do, perhaps that says something about our capacity to control or get desired outcomes within biological systems. So, and again, this gap between in vitro and in vivo is it's a vast one. Modifying human behavior. So if you think of the big healthcare picture, but also trying to change processes within organizations, within academic organizations where we still have certain models of doing things that go back centuries. The change management is pretty difficult. So there's a very interesting guy named Richard Hamming, famous for Hamming codes, and he gave a talk about research, and he says, do you want to spend your time working on the system, or do you want to be a first-class scientist? It's very difficult to do both. And I thought a lot about that. I've been seeing the system and diagnosing its things, but not necessarily being in a place where I could have control over it, right? And so the question is, do we spend all our time, bitching about the system, or actually do something at the level where we can actually influence it? So I do like to look at the system, and if you will, by looking at many sort of academic organizations, been able to draw general principles, and then not say, well, they all need to change. Well, no, but if I'm going into academia, for my local system, what can I learn? And so I'd like to talk about some of the observations and things that I've seen. And then ideally we could, not spend your life bitching about the system, but then work on having an excellent system ourselves. So I did publish this article in Biostatistics where I was looking at the patterns that we were seeing in GWAS studies and sort of some systematic problems. And the question is, why should I care about my research process? Well, if you think of what we're doing in academia, we're trying to generate knowledge and find significant findings. That's what tends to drive us. We're curious people. But we operate in a system that's really driven by reputation, and publication is one key measure of that, but also the grants we get and so forth. And there's a virtuous loop where if we can find some really good stuff and get great publications, our reputation goes up, we get more funding, we get more publications, we can build more research capacity to do more significant findings, and much like companies grow, research organizations can grow and be very successful. We all know there's some luck involved in that. You can get lucky with your reviewers, lucky with your... and then the research competence is really the one thing that we have control over. And so thinking about not just working in the system, but on the system, our own personal research process, I believe is the real leverage point for getting the most, we have limited time, limited budget, how do we get the most out of it? So we could try to bring all these business concepts in, but I thought the best way to frame it as scientists is to say we need to be scientists on our research process as well as in our research process. And so it's really about that OODA loop, if you will, I got to form a mental model of my research process and have hypotheses about what works and doesn't work and try some things out and rinse and repeat. And so I set out sort of a postulated better way of doing drug discovery. Could be wrong, it may not be sufficient, but it might give an improvement and I could learn from it. And then some keys for success that often are missing, especially in a world where the things we're measuring are kind of murkier than dollars and cents in business. So in business, it's very clear you want to increase shareholder value, more money per time, right? In a research system, it's meaningful knowledge. But we kind of have sub-metrics that, reputation sort of a proxy. If that person did good things and his or her peers said it was good, that was probably meaningful knowledge. And so we have reputation units per time, grant dollars per time, commercialization dollars per time if we're trying to look at other ways of funding our research. And then there's our investment and our operating expense that we also have to pay attention. So the question is can we have a way to focus on those levers for bioscience research and get more? So some of the levers that and I'm not the first to point them out, but we're all familiar perhaps with, maybe not all, but the Begley and Ellis paper from Nature in 2012, and there's a couple other ones showing that reproducible research is a real problem. So these guys at Amgen were wanting to do cancer drug discovery, and they were looking at 53 different landmark, top-tier journal publications on preclinical cancer stuff. And so the peers said this was great and said, hey, this could be a great starting point for our research. They worked many times with the authors to try to reproduce, the findings, and only about 1 in 9 studies were they able to reproduce it, even getting the help of the original authors. Now, we know from the scientific method is we look at specific instances in reality, we form conjectures about a general case, and then we'll do a test on more specific cases to see if they falsify that generalization. If we're in a situation where our research is not refutable, and this is a problem when they talk about reproducible research, we're not making the data available, or we're not trying ourselves to refute our work. And so this is the real problem, is in a system governed by reputation, getting something exciting and new out there actually enhances all those feedback loops that I talked about, but actually enhancing meaningful knowledge that we can build on takes a very rigorous reputation. And so again, this idea that if the front of the process is where the errors are introduced, so subsequent to this 2006 publication of mine, I thought, if 8 out of 9 of the top flight stuff, maybe it's not that bad, but it's probably not 50% reproducible. If there's more non-reproducible, non-solid stuff than good stuff, think how that must be amplifying through our whole drug discovery and research process for trying to understand disease and so forth. Seems like a huge leverage point. So come back to this idea that the prime metric of academia is reputation. Right? In business, it's money. In academia, it's actually reputation. And so knowing that that's the goal units of the system that we're trying to thrive in, is one thing. we could look at the flaws of it, though. I mean, it's really a shortcut to making decisions. Because how the heck do I say this guy, Christophe, knows what he's talking about? Well, his peers said he's good, he's went to a good school, whatever. But do we really have a mechanism of assessing the actual, and it's a question of even the long-term implications of, that there was a gal who got the some Nobel Prize in chemistry, I believe, and her publications were hardly cited until after she got the Nobel Prize. So to truly assess the actual, academic value of something, reputation's just a proxy, but it's the proxy we use. But the problem with this proxy, as I say, is it undermines the scientific method, because it encourages data dredging. And this is what we've seen in so many publications. Find a finding, search for corroboration rather than falsification, tell a nice story, publish the paper, and move on to finding another correlation. And this is falling short of really good causal understanding of the types of models that I was talking about earlier. And so there's some ways that we as scientists can get out of this. First, there's a psychological thing with humans. We're probably not wired very well to follow the scientific method. We like to be right. We like to find information that corroborates and backs ourselves up. And so back in 1890, this guy Chamberlain authored in Science this article. It's been reprinted since in Science in the 60s, and it sort of gets rediscovered every half century or so. But he says, the problem is we have this parental affection for our hypotheses, right? And so one way to sort of minimize that is have many parallel hypotheses that we already know, many of them are going to be struck down, so already we have less attachment to them. And then we can form a series of experiments to strike down whole swaths of hypotheses. And in Platt, who wrote a commentary when this was reproduced in the 60s, he was describing, Watson and Crick, he was describing Crick's blackboard of how they were basically building these kind of decision trees of hypotheses and chopping whole swaths of the trees down, and they were following this approach. And I look at myself and I look at many of the colleagues, and it's like, where have we lost this sort of rigorous formulation of our decision tree of hypotheses and so forth, and be very objective about it. And so maybe some of the drop we've seen in research productivity is perhaps a loss of a certain mindset that used to be with us in a system that's so reputation oriented and has only gone up in that. Maybe we're losing some of that. And in my paper on learning from our GWAS mistakes, we saw some top statistical mistakes that it really comes in that multiple testing, overfitting, and the biggest thing that is design of experiments gets ignored. The Wellcome Trust, which published its first big GWAS on 7 different disease areas and sort of got the area going. They collected all the controls from once. Well, they had 2 sets of controls from 2 different sites, and then the cases were picked from different sites. And then they ran all the controls on one batch of plates, and then they ran the cases. Sometimes they mixed the cases on plates, but they didn't have any design of experiments where you want a block randomized design of cases and controls on the same plate, so that you're not introducing basically complete confounding twice. And so I had the chance to work on about 30 different GWAS studies, and there was only 2 that had a proper block randomized design. One from GlaxoSmithKline, where they had good clinical trial statisticians who designed it very carefully, and then one that we designed on behalf of Heidi Piami at the Wadsworth Institute. And when you do the statistical analysis on those 2 studies, this whole problem with so-called bad SNPs or that we've got to drop all sorts of data and build rules for throwing out outliers. Those outliers were all due to that bad experimental design, and there's been retractions based on this and so forth. And so tens to hundreds of millions have been spent on experiments where, not that they didn't get something out of it, but if proper design of experiments had been done, we wouldn't need a whole statistical cottage industry trying to go correct for that, and in many cases you just simply can't. So advocating having either teaching it to yourself or learning it, it's really, I learned it in a week course at Glaxo, Welcome Design of Experiments, is an enormously powerful leverage point to not be wasting, especially money on these incredibly expensive experiments. And so much as there's this feedback loop in improving productivity for in terms of generating reputation and grants and so forth, you can look at the analytic competency and capacity. Science has moved to becoming a heavily data-oriented and analysis-oriented activity, and what's kind of happened is there's been a generation gap where if we go back to 20 years or so, the senior scientists were the most complex analysis was maybe a little bit of spreadsheet work, and the big data stuff that we're doing today, those scientists weren't doing it 20 years ago. So the mentorship of the younger generation on how you do proper design of experiments in the context of analysis of big data has kind of been lost. And so, you can just look at this graph, but the gist of it is everything from design of experiments to statistical analysis and so forth. If we've seen people sitting 2 years on data sets, unable, they spent $1 million to run an experiment, and then they buy Golden Helix software, and even a salesperson would actually get them further along in their project in one afternoon than they had in 2 years, where they were often waiting for what they call the high priest of bioinformatics to actually do their analysis. And so we can see for the haves and have-nots in terms of analysis, it either is a great reinforcing cycle to have it, or if you have crappy analysis, you can be talking retraction and embarrassment and basically killing your reputation, which I spoke about a notable case in a webcast about a year ago, where it was just the graduate student, the inmates were running the asylum. The senior professor thought, they did the analysis, it came with the results. They didn't have the ability to sort of assess these issues of both experimental design and statistical analysis, and ran into problems. So the other biggie is you think about, I think about moving from industry to academia. And in industry, if we run out of money, we don't have sort of that tenure position to fall back on. We lose our jobs. And so we build sales funnels to manage the flow of deals and money. And so I did a little simulation on grant funding and said, well, if I submitted an SBIR grant or a R01 grant, and the interesting numbers are kind of similar in terms of, and you look at the success rate on the first and second tries, and you resubmit the ones that fail, and you build a simulation model, and you look at the lower and upper bounds, the 95% confidence interval of how either good or bad it could be, which is the green lines. If I was to start, this was looking at getting the first August deadlines going, and submit 3 a year plus all the resubmissions. It could take till 2015 where you could still have no money. You could just be unlucky, right, in that lower 5%, but you'd be pretty certain by 2016 you'd have some money. So you could also be very lucky. So this variability for an individual researcher, even at a fairly prolific rate of generating, it taps out about six proposals a year that you're the PI for. But if you were to take a group of 10 professors, if they were all writing grants together and sharing in the wealth, the variability goes down dramatically, and you can actually get money a lot sooner, and you even it out. So if we look systemically at being an individual researcher, the variability we're subject to, no wonder it's such a stressor on all of us. And so thinking about, as a department organization, can you do some sort of spreading of that variability or reduction of the variability, but B, also the managing of that pipeline and that funnel. So many times it's, oh crap, I ran out of money, better write grants. But if we look at the potential lag of starting from nothing, we could be unlucky and wait 2-3 years to start getting money again. So the actual management of our livelihood, something we're not taught in graduate school, right, as biologists or chemists or statisticians or whatever, and yet as a business person, I was forced to learn process and metrics and so forth on all this. We often think business is, and for those of us driven by curiosity, all that process stuff is like killing us. So then it becomes, can we get really capable administrators, not secretaries or people who can just do budget, but process excellence people to assist us in basically managing, our livelihoods. And unfortunately, it's almost absent. You'll see it, in some very top flight universities where they take seriously that, but it's a real challenge. So another thing that it's interesting, there's not been a lot of citation of this article in Science, but there's a group out in Scotland and in the UK that has basically built a instrument where they could automate tens of thousands of experiments and essentially turn. So my fantasy as a computer scientist going into research would be, from your keyboard, you could essentially design your research process and start running it and have automated inference going on, automated running of the actual instruments and so forth. And we're not there yet, and there's some early things going on, but to me, as I see this convergence of computer science and science, to me it's inevitably going towards this automation where more and more I think we're going to be less doing the pipetting by hand and going to a place of research automations, because really think about the rate limiting step. We're trying to probe this vast complexity, and that also leads me to the idea of automation of inference, and one of the reasons I'm teaching this semantic web course at Montana State this semester is there's a whole new set of technologies that is to do for the web what the web has done for sort of text and information sharing with data. So how do we connect all the data in the world? And so that's coming in the next few years, and how do we get chemistry and genetics data sets, but also public data sets from the NIH in a format where it's, as most people who do data analysis know, 90% of their time is spent futzing with data, and 5% is actually doing the analysis, and so there's a real power of automation inference that's going to be coming. Another sort of systems thinking discipline that I would just, as a tantalizing thing, throw out there, not go into what it is. There's something called TRIZ, a Russian inventive system, or it's actually pronounced TRIZ, and it's this Russian acronym. But it is actually, we get into blind alleys in our thinking, and there's a way to soup up our innovation capability. And there's an interesting study done here where a bunch of people, hundreds of people, were given the same creative solving a problem to solve. Only 2% could get a solution just thinking, as we all do. We think we're smart, and with 1 week of training, they were able to get 68% of a different population able to solve the same problem with this inventive training. So if you think of, we're all about invention, And yet there are some things out there we've never heard about. Apparently there was a Russian inventor who had 230 patents or something like that, or 200 and something patents. And he was exposed to this Altshuler's TRIZ methodology, and he wept. Because he said, here I'm at the end of my career, and if I had known at the beginning what I know now, and what could I have done with my life? Bad multitasking is another thing. So we're beasts driven by curiosity. We also learn through synergizing across different things as scientists. And that tends to drive us to multitasking across many areas of interest. But if we actually want to get something done, as demonstrated by this graph, if you have 3 tasks and you just do one after the other, this one's done here, this one's done here, this one's done here. If we do a third of it and then switch, and then a third, and then switch, and a third, and switch, the first task is done at time point 35 here. So when we think of the ROI of producing reputation and so forth, and we got a massive set of projects going on, we're pushing out the actual getting the reward of it until later. And then it's worse than that because there's context switching. You forget what you're doing. You got to get yourself up to speed. You get out of your flow mode on one thing. And so the truth is, and who of us has 3 things only going, right? Isn't it more like 20, right? And so the surprising, and it comes from a childhood thing, I think, which is, if you want to get something done, you better get it started now, which is absolutely true for one task, but absolutely false for the larger system of many tasks. And yet, actually shifting our mind to being focused on one thing or a few things. Not to say you should no multitasking. It's when it gets to a point where it's too bad. And so there's a right place, and we probably err on the side of too much. And just cutting it back a bit should boost our productivity. How do we focus? There's high-value projects that are easy to do. We'll call them pearls. We should do those first. There's ones that are high value, but we got to pry the pearl out of the oyster. We should do those. And there's low value things that are easy to get. But then there's low value, high effort things. And just a simple prioritization, thinking about the ROI of the projects we're going to get and how much work it takes to get them done is an enormous focusing thing. We can typically cut out 25% of the projects we're doing right off the bat and 25% of the scope of the projects that we're continuing to do, where it's just stuff that doesn't need to be done. So, this quote becomes cliche that insanity is doing the same thing over and over again, expecting different results. But look, we've thrown a lot of money at all sorts of problems like this, and we've thrown a lot of smart people at it and think, well, I'm going to spend more, or I'm going to be smarter than everybody else doing the same thing, and we're going to get a difference. I think we need to rethink and start thinking about our system and work not just in it but on it. And none of us are getting any younger, and so this is all going to become all too personal at some point. And so, wouldn't it be nice if we could get 1 year of life for every 1 year of research, and then we'd live forever, right? But hopefully, we at least can make some improvement in that and if not for ourselves, for our children. Thank you.
Yeah. Go ahead. Questions for?
Yeah, so I've got a question, more of a comment though.
Yeah.
Because I think 2 things you touched on very astutely. First of all, very nice talking about it. You talked about reputation and how that drives, science and success. And then later on you talked in the model of individual versus collaborative efforts to try to get funding.
Yeah.
And the comment I have is just something I've said to DJ before. I've noticed increasingly now with NIH study sections when I go and review these, training grants or program grants or whatever, what used to start as sort of a university-specific application. So University X puts in this, and they're competitive. Now it's like the Walmart phenomenon. You get these monster consortia that are forming.
Right.
We see this in global health, the big training grants that they have.
Yeah.
5, 10 universities, and you get these packages where they'll take every D43 and T32 that they have in the institution, or the group of them. There could be hundreds, and just stack them all up. And really, they're leveraging the reputation now of the
Institutions
consortiums.
and the consortium. Yeah.
And they're actually, if you will, putting this massive group effort in to maximize their chances for the one grant that's out there.
Yeah.
And so I think that's a very astute,
Yeah.
point that you really do.
Yeah, so it's individual reputation, it's department, it's institutional, and then it's, as you say, multi-institutional reputation. So it's kind of a thing to think about too. Is in addition to managing your grants funnel, if you will, there's a brand management that one has to think about, because you don't just get reputation by getting the cover of Science or Nature or whatever. Self-promotion is, it's brand management. So it's funny at Golden Helix, I do these webcasts, and even some of my head of development and so forth, we'll get 400 or 500. We've had 750 people sign up for a webcast. So it's like filling and then we'll get 50, 60% show up. So you have several hundred people. Then you go to a conference, you're like, oh, you're famous. They'll say that to my head developer. He's like, cool. So I record these things because it's actually part of building one's personal brand identity. And am I a guy who really cares about being famous? No, I'd love to do great science and so forth, but recognizing that that's the system we're in, and the wallflower doesn't necessarily get the funding. So how many departments have an actual head of marketing? Which is kind of what goes in, and it's, this is dirty, this is, I didn't create the system.
Yeah, the marketing department's the only one.
If we just had an objective measure of scientific contribution, but we don't. And so that's the system we work in. Other questions or comments?
There's a few parallel systems. One of them is from Iconics, which they collected about 800 molecules, and then they tested them on 100 plus pharmacological assays and many more microarray assays.
Mm-hmm.
And then that data, so they sort of went bankrupt with
Yeah.
this. They were acquired by Entelos. At some point, Entelos were charging $50,000 just to have a look at the data.
Yeah.
And then
Yeah.
that
NovaScreen
didn't
was
work
trying to
out.
charge for a look at that data.
And so that data is now publicly available through the National Toxicology Program.
Right.
NTP.
Yeah.
It's called the Drug Matrix.
Yeah, it's still kind of small though, right? 800. And to your point the other day about
It is
the generalization beyond that sample size of 800. So what you really want is miniaturization and so forth, and having the ability to do these hundreds of assays, in a small space, cost effectively. That's what's never been done, right? But if you were to think, like you work on all sorts of instrumentation stuff, and we're doing incredible on throughput and cost for genotyping, but for all these assays, the flow cytometry assays and so forth, we don't have kind of an equivalent high throughput for
So
hundreds of assays.
In many ways, that was the intent with the molecular libraries, which was get your hands on 400,000 molecules and screen them across multiple assays.
Right.
And in a way that was done, but unfortunately, by the time they actually got the library and they started to do it in earnest, they had already produced a lot of assays that didn't include the whole library. And then there's a lot of random variability per assay. So that there's a lot of
A lot of noise in that data.
mishap
Yeah.
that's related to this. But
So-
that was the intent, to create this space of
Mhm.
chemical biology.
Right. And what I was representing too was, when you think of innovation, it's good to picture at least, your ideal system. Ideal system be free to screen an infinite number of compounds across, an infinite number of, or as many biological systems as there are. And then you say, okay, well, I can't do that today, but if I was to gear my research towards closing that gap, then, maybe in a decade we'll be there.
Any other questions? Thank you all for coming.
Thank you.